Usually when we talk about getting a medical diagnosis, there's this expectation of absolute precision, like engineering almost.
Right, yeah, like a binary thing.
Exactly. Like if you fall off a ladder and break your arm, the x-ray just shows that stark, jagged white line. The doctor points at the screen and says, there it is. Broken or not broken, it's clean, definitively clean.
But if you suspect something is wrong with your brain, right, like your memory or your cognition, historically it hasn't been like that at all.
Not even close.
No, it has been much more of an educated guessing game. I mean, for decades, if a patient at a memory clinic came in, they'd undergo these subjective cognitive tests, maybe draw the face of a clock, answer a questionnaire.
Yeah, the classic tests.
Right, or if they had the resources, they'd get an incredibly expensive radioactive PET scan. But that was rare.
Which is exactly why today's deep dive it's so fascinating. Because our mission today is to completely decode a groundbreaking 2026 Nature review.
Yeah, by Henrik Zetterberg and Barbara B. Bendlin.
Right, exactly. And this paper is entirely about biofluid biomarkers in neurodegenerative dementias. We're taking this incredibly dense stack of scientific literature and essentially translating it into a guide on how tiny drops of blood or spinal fluid can now reveal the microscopic molecular secrets of your brain.
It's revolutionary.
It really is. Imagine you were sitting in a clinic, right? What if a simple blood draw could tell a doctor exactly which proteins are misbehaving in your brain long before you ever, forget where you put your keys?
Because we're shifting away from that whole paradigm of symptom-based guessing, we're moving to what the medical field calls molecular phenotyping.
Molecular phenotyping.
Right. So instead of waiting for the disease to cause outward physical damage, we can now use fluid from the cerebrospinal fluid or CSF or blood plasma to literally map out Alzheimer's, Parkinson's, and other dementias decades before the first symptoms ever appear.
Decades. That completely changes the timeline of human health.
It really does. It's a total paradigm shift.
Okay, so let's unpack this. If we're going to trace these molecular footprints, we have to start with the most famous ones, right? The classic Alzheimer's triggers, amyloid-β and tau.
The big two.
Yeah. And I've always thought of these two as like the sticky trash and the collapsing scaffolding.
Right.
So let's look at the trash first. We hear about amyloid plaques all the time, but what exactly is amyloid-β or A-beta doing on a microscopic level?
Well, amyloid-β is actually simply a normal protein fragment. Specifically, it's a 42 amino acid long peptide. In a perfectly healthy brain, your cells produce it, they use it, and then clear it away like normal cellular exhaust.
Like taking out the trash.
Exactly. But in Alzheimer's disease, this specific fragment, a beta 42, it starts to misfold and it clumps together. So it forms these extracellular plaques, literally just piles of sticky trash sitting outside the neurons, disrupting their ability to communicate.
Okay, but here's a detail from our sources that initially threw me off. If you have Alzheimer's and your brain is filling up with these A-beta 42 plaques, the concentration of A-beta 42 in your spinal fluid actually drops.
Right, it goes down.
Which is so weird, because if the trash is piling up, shouldn't there be more of it floating around in the fluid?
Yeah, it seems totally counterintuitive until you look at the actual fluid dynamics of the brain. The A-beta 42 concentration is dropping in the spinal fluid precisely because it's acting like a sponge in the brain tissue.
Oh, I see.
Yeah. The protein is getting trapped in those sticky, insoluble plaques. It's locked up. So it literally can't freely wash out into the spinal fluid.
Wow. Okay, that makes sense.
But, and this is important, just measuring a low level of A-beta 42 isn't enough to confirm a diagnosis because everyone is different. Doctors actually look at a very specific ratio. They compare A-beta 42 to another, slightly shorter fragment called A-beta 40.
Right, because if you just measure the total amount of trash, you don't know how big the house producing it is.
Right.
It's like some people naturally produce more protein at baseline than others.
Exactly. So the ratio normalizes for your personal baseline of protein production and clearance. When clinicians use that A-beta 42 over 40 ratio in spinal fluid, the result is almost 100% concordant with an expensive PE scan. It's remarkably accurate.
That brings up a massive question, though. The whole point of this new era is moving away from painful spinal taps to simple blood tests, right?
Yep, that's the holy grail.
Yet the sources note that the A-beta signal in the blood is frustratingly weak. We're talking about only a 10 to 15% change in the blood compared to a massive 50% drop in the spinal fluid. So why the massive drop-off in accuracy?
Well, blood introduces a severe masking effect because, your brain isn't the only organ producing amyloid-β. Extracerebral sources, particularly your blood platelets, produce their own beta.
Oh, of course.
Right. So when a phlebotomist draws your blood, they are capturing this chaotic mix of brain amyloid and peripheral platelet amyloid. All that background noise just muddies the waters, making the crucial drop in the brain's beta much, much harder to detect.
Okay, so the sticky trash test has a major background noise problem in the blood, which brings us to the second classic protein, tau. And if amyloid is the sticky trash piling up outside the house, tau is the collapsing scaffolding inside the house, right?
That's a perfect analogy. Tau is a fascinating protein. Its normal day job is to stabilize microtubules. Think of microtubules as the internal scaffolding and the highway system inside the long axon of a nerve cell.
Okay, holding the shape together.
Exactly. But in Alzheimer's, tau goes through a process called hyperphosphorylation. It gets chemically loaded with way too many phosphate groups. And those phosphate groups act like a signal, causing the tau to physically detach from the microtubules.
Oh, no.
Yeah. The internal scaffolding of the neuron literally collapses. And that detached tau then tangles up on itself, creating what we call neurofibrillary tangles inside the cell.
Okay, and the sources mention that we can measure this in the blood now using specific markers. You'll see them listed as p-tau181, p-tau231, or the current leading candidate, which is p-tau217. But there is a massive caveat here about what these p-tau blood tests are actually telling us, isn't there?
There is, and this is perhaps the most misunderstood part of the new blood tests right now. When you measure p-tau in a patient's blood, you are primarily measuring the brain's real-time reaction to amyloid-β.
Wait, really? It's a reaction to the amyloid?
Yes. Neurons get stressed by the surrounding amyloid plaques, and they start overproducing and secreting this phosphorylated tau as a stress response. So a high p-tau blood test does not definitively mean the brain is filled with fully formed mature tangles. It just means the brain is panicking.
So wait a minute. If someone has a disease that causes tangles but doesn't involve amyloid plaques at all, does the test fail?
Completely. Yeah, we see this in primary tauopathies. These are diseases like certain types of frontotemporal dementia, where tau tangles form independently of amyloid. In those patients, standard p-tau blood tests will often come back totally normal.
Even while their brain is physically tangling up.
Exactly. Because the amyloid trigger isn't there to force the secretion of the p-tau.
That is wild. So to see the actual physical tangles, regardless of what caused them, the researchers had to find a different marker entirely.
They did. They had to look for fragments from the microtubule binding region of the tau protein itself. In spinal fluid, this highly specific marker is called MTBR tau243. And recently, scientists isolated a version that spills into the blood called eMTBR tau243.
And what does that do?
Well, that specific fragment is believed to break off from the fuzzy outer coat of the mature tangles themselves. So measuring that gives doctors a direct window into actual tangle pathology. It completely bypasses the amyloid stress response.
Okay, so just to make sure I'm tracking this molecular detective work right, a quick summary. 1, we can't fully trust a simple blood amyloid test because our blood platelets throw off the numbers. 2, the most famous blood tau tests like p-tau217 are really just showing us the brain panicking over amyloid.
Right.
And 3, MTBR tau243 shows the actual tangles, which makes me wonder, as a patient navigating all this, would you insist on getting the more invasive spinal tap just for the absolute certainty of that clear A-beta ratio and MTBR marker, or would you be comfortable starting with the slightly murky blood test?
Well, that risk versus reward calculation is going to be the central conversation in neurology clinics for the next decade, honestly.
I bet, and it perfectly sets the stage for the next piece of the puzzle. Because amyloid and tau are just the triggers, right? They are the match and the kindling. But if we want to know if the brain is actually burning down, we need to look at the dying cells themselves. How do we measure actual brain damage?
Right, so here we pivot from pathology markers to markers of neurodegeneration. And the undisputed heavyweight here is a protein called NFL, or neurofilament light.
The ultimate check engine light for the brain.
Absolutely. NFL is a structural protein that lives deep inside the axons of your neurons. If a neuron sustains critical damage and dies, whether that's a slow death from Alzheimer's, a sudden death from a stroke, or blunt force trauma from a concussion, the cell membrane bursts open.
And it just spills out.
Exactly. It spills its internal contents, including NFL, directly into the spinal fluid, which eventually filters into the blood. So if your blood NFL levels are rising, it is a binary signal. brain cells are actively dying.
And this isn't just an abstract theory either. The sources detail a massive real-world application of this. The FDA recently approved a breakthrough ALS drug called tofersen.
Yes. A huge milestone.
And the defining reason for approval was that the drug drastically reduced NFL levels in clinical trial patients. The FDA basically looked at the data and said, hey, the check engine light turned off, the neurons stopped bursting, so the drug is doing something.
It was a watershed moment. It totally validated biomarkers as clinical endpoints, but relying solely on NFL presents a significant specificity issue.
Oh, why is that?
Because NFL exists in your central nervous system, but it also exists in your peripheral nerves. You know, the nervous system controlling your arms, legs, and organs.
Oh, so if you damage your leg.
Exactly. If a patient has a severe peripheral neuropathy, perhaps from diabetes, their blood NFL will spike dramatically, and it has absolutely nothing to do with dementia.
Which leaves you in a tough spot as a patient, right? If your blood test shows your check engine light is flashing, you're immediately going to ask your doctor, okay, but which part of the engine is actually broken? My brain or my legs?
Right. And researchers are actively solving this right now by developing an assay for a protein called alpha-internexin. Unlike NFL, alpha-internexin is a neurofilament that is exclusively found in the central nervous system.
Nice.
Yeah, so by comparing the two, doctors will soon be able to definitively trace the origin of the cellular death. And we are actually seeing this exact same refinement process with tau, too. General total tau gets mixed up with peripheral tau in the blood, so scientists engineered highly specific antibodies for brain-derived tau, or BD tau.
So it essentially mirrors the accuracy of a spinal tap, but from a simple arm draw.
Precisely.
Okay, so that covers the neurons dying. But anytime there is a fire, a cleanup crew shows up. How do we measure the brain's immune system freaking out?
We look at glial reactivity. Astrocytes and microglia are the dedicated support and immune cells of the brain. When they sense cellular debris or invasive amyloid plaques, they activate. They physically change their shape and start releasing distress proteins. In the spinal fluid, we measure markers like YKL40 and STRIM2.
But the real standout for blood tests here is GFAP, right? Glial fibrillary acidic protein.
Yes. GFAP is strongly expressed by brain astrocytes. When we measure GFAP levels in blood plasma, it correlates incredibly tightly with amyloid-β pathology. It serves as a direct readout that the astrocytes are mobilizing to fight the plaques.
Okay, so to synthesize all of Section 2, NFL acts as the general alarm bell that cells are dying. Emerging markers like alpha-internexin and BD tau act as location trackers, so know the damage is definitely in the brain. And GFAP shows us the astrocyte cleanup crew mobilizing in the blood.
Spot on.
But here's a thought exercise for you listening. If a routine lab panel showed your NFL alarm bell was ringing loudly, but your amyloid and tau were completely normal, what on earth is killing the brain cell?
That's the million-dollar question. Because if we connect this to the bigger picture, Alzheimer's rarely acts alone. Most older adults have mixed pathologies, which is why a purely Alzheimer's-focused diagnostic lens is actually quite dangerous.
And here's where it gets really interesting. Our sources dive deeply into two other massive villainous proteins, alpha-synuclein and TDP-43. So let's start with alpha-synuclein, which is the protein responsible for Parkinson's disease and Lewy body dementia. What is the mechanism here?
Well, alpha-synuclein is typically a totally benign functional protein located at our synapses, but in diseases like Parkinson's, it begins to misfold and clump together into these aggregates called Lewy bodies.
Okay.
The diagnostic hurdle for decades was that measuring the total amount of alpha-synuclein floating in a patient's fluid was entirely useless, because healthy brains and Parkinson's brains have largely overlapping amounts of the normal protein.
So if counting the total number of proteins doesn't work, how do we hunt down the tiny fraction of misfolded ones?
Scientists developed seed amplification assays, or SAAs. The mechanism relies entirely on the misfolded protein's insidious prion-like behavior.
Kind of like the bad apple ruining the bunch.
That analogy is biologically accurate here, yeah.
Yeah.
If you have a barrel of healthy apples and you introduce one rotting apple, the rot spreads by physical contact. Misfolded alpha-synuclein acts the exact same way. When it physically touches a normally folded alpha-synuclein protein, it forces that healthy protein to misfold as well.
Wow.
Right. So in an SAA test, technicians take a tiny sample of a patient's spinal fluid and place it in a test tube filled with normal, fluorescently tagged alpha-synuclein.
And they shake it up?
They shake it and they wait. If the patient's fluid contains even a microscopic seed of the pathological protein, it triggers a chain reaction. The misfolding amplifies exponentially, turning the entire test tube pathological, and the fluorescent tags light up.
That is just brilliant. It detects the disease with staggering accuracy by literally letting the disease reveal itself, finding the bad apple by letting it rot the test tube.
It's an elegant solution.
So cool. Now let's look at TDP-43. This is the protein behind ALS, frontotemporal dementia, and a surprisingly common disease called late. And late mimics Alzheimer's memory loss so perfectly in the elderly that it's constantly misdiagnosed.
It is this all the time. So TDP-43 normally resides inside the nucleus of the cell. Its primary job is RNA regulation. If you think of the nucleus as a library and RNA is a photocopied blueprint of a gene, TDP-43 is the editor. It ensures the blueprint makes grammatical sense before it gets sent out to build a protein.
Okay, I like that, the editor.
Right, but in diseases like late, TDP-43 escapes the nucleus, it clumps up in the cytoplasm, and basically abandons its editing job entirely.
But the sources specifically warn that we cannot just measure this rogue TDP-43 in the blood. Why not?
We can't, because normal TDP-43 is completely ubiquitous in the human body. It is especially concentrated in red blood cells. So if a phlebotomist accidentally pops even a single red blood cell during a blood draw, millions of normal TDP-43 proteins flood the sample, instantly ruining any diagnostic signal.
Oh, wow. So we can't measure the broken editor? How do we know it failed?
What's fascinating here is that the scientific community sidestepped the protein entirely and looked at the consequences of its absence. Because TDP-43 isn't in the nucleus editing the RNA gets misspliced. It retains these random chunks of genetic gibberish that were supposed to be cut out.
The typos.
Exactly. We call these retained chunks cryptic exons. The cell reads this gibberish blueprint and starts manufacturing bizarre, novel proteins that should not exist in a human body at all, like a specific peptide string called HDGFL2CE.
I love this. We can't measure the broken editor, so we just scan the blood for the typos it left behind. We find that HDGFL2CE typo, and we instantly know TDP-43 has failed.
It's a true masterpiece of lateral thinking in biomarker design.
Okay, recap time. We've got seed amplification assays acting like biological amplifiers to catch Parkinson's, and cryptic exons acting as typo detectors to catch late and ALS. I want you to think about the gravity of this for a second. Knowing that late perfectly mimics Alzheimer's, how do you feel about the reality that thousands of people currently diagnosed with Alzheimer's might actually have a completely different molecular disease?
It's a sobering thought, because giving an Alzheimer's drug to a late patient is biologically useless. These specific biofluid tests aren't just cool science, they're literally the only way to get the right treatment.
It fundamentally reshapes how we design clinical trials too, right? We can finally ensure we're testing the right drug on the exact right molecule.
Exactly. But tracing these big famous proteins isn't the whole picture. We need to know the state of the brain's actual hardware too.
Right. The wiring, which are the synapses, and the plumbing, which are the blood vessels.
Yeah. So let's examine the wiring first. Synaptic dysfunction is arguably the earliest physiological event in cognitive decline. In the spinal fluid, we track markers of synaptic distress. A postsynaptic protein called neurogranin reliably spikes in Alzheimer's patients. We also see presynaptic markers. These are proteins involved in releasing neurotransmitters, like SNAP25 and 1433. They spike in the fluid too.
Wait, I want to challenge that logic for a second. If synapses are dying and disappearing, shouldn't the proteins that make up those synapses decrease in the fluid? Why are they spiking?
It's a brilliant question. The spike represents a pathological hyperexcitability early in the disease process. Before the neurons die, they essentially short-circuit. They fire erratically and excessively, dumping massive amounts of these presynaptic proteins into the fluid.
So it's a short circuit before the blackout.
Precisely. But there is a synaptic protein that does drop, which is NPTX2.
NPTX2.
Yes. NPTX2 is a transsynaptic protein. Its levels actually decrease because its secretion is tied directly to intact functional synapses. As the physical synapses are finally destroyed, less NPTX2 is produced. Researchers realized that by creating a mathematical ratio dividing the spiking SNAP-25 by the plummeting NPTX2, they could capture both the erratic short-circuiting and the physical loss simultaneously.
That's incredibly clever.
It is an incredibly powerful predictor of imminent cognitive decline.
And researchers are finally getting synaptic signals in the blood too. The sources highlight beta-synuclein, which is highly enriched in central nervous system synapses and can be measured from a simple blood draw to show early synaptic fraying.
Yes, that's a huge step forward.
Okay, so what about the plumbing, the cerebrovascular system?
The plumbing markers depend entirely on the speed of the failure. If a patient suffers an acute stroke, a massive sudden plumbing failure, you see immediate violent spikes in GFA and t-tau within hours, followed by a long, slow burn of NFL over the following weeks.
And what if it's not acute?
Well, chronic smoldering small vessel disease is stealthier. The markers are still emerging, but researchers are tracking SPDGFR-beta, which is a protein shed by the parasite cells that wrap and protect blood vessels, or they are looking at placental growth factor in the blood plasma.
Okay, so we can track the fraying wiring with the S-anzanep-25 ratio and beta-synuclein, and we can monitor the plumbing for acute blowouts or slow leaks. Imagine getting a lab result showing your brain's wiring was fraying years before you ever noticed a memory slip. What lifestyle changes, what interventions would you immediately make? It's the ultimate early warning system.
It is, but an early warning system is only useful if it doesn't give you false alarms.
Right. So what does this all actually mean when these hyper-advanced tests leave the pristine research lab and hit your local doctor's office? Because this molecular phenotyping is not completely foolproof, is it?
No, it has serious vulnerabilities. The most critical pitfall is patient comorbidities. Take chronic kidney disease, specifically a condition called shrunken pore syndrome. If your kidney's filtration system is impaired, they fail to clear circulating proteins from your blood.
Oh, so they build up.
Exactly. This causes plasma levels of p-tau and NFL to artificially skyrocket. A patient's brain might be structurally perfect, but their failing kidneys create a biomarker profile that screams Alzheimer's. Even an extreme body mass index can significantly alter NFL baselines.
So the doctor can't just blindly read the lab sheet. They have to treat the whole patient. There's also a major risk with those mathematical ratios we praised earlier, right? The ratio trap.
Yes, the ratio trap. Ratios are powerful, but mathematically they amplify any underlying assay error. For example, if a patient is prescribed Entresto, which is a very common heart failure medication, that specific drug artificially increases plasma A-beta 42 levels.
Oh, no.
Yeah. If a clinician unknowingly plugs that artificially inflated a beta 42 number into a diagnostic ratio, the entire result inverts. The diagnosis is completely ruined by a cardiac pill.
That is terrifying. Then there is the sheer physical handling of the blood, what we call pre-analytics. If a busy nurse leaves the blood tube sitting on the counter at room temperature for an afternoon, the delicate neuroproteins degrade. But the sources reveal a brilliant, elegantly simple workflow hack for clinics so they don't have to invent new protocols. They call it the homocysteine trick.
It really is a stroke of logistical genius. Global clinics were terrified they would need expensive, specialized training to handle these new brain biomarkers. But researchers realized that hospitals already possess a strict, time-sensitive protocol for drawing routine vitamin B12 and homocysteine tests.
Right, something they do every day.
Exactly. The protocol is simple. Draw the blood into an EDTA plasma tube, put it directly into a centrifuge, and separate the plasma within exactly 1 hour. If clinics simply piggyback on that existing homocysteine routine, the Alzheimer's biomarkers remain perfectly stable. No new training, no millions of dollars in logistical overhaul. Just use the system already in place.
It's amazing how a massive scientific breakthrough ultimately relies on the centrifuge habits of a local clinic.
It really is.
So we have to watch out for kidney false positives, medication interference like Entresto, and the clock ticking on the blood tube. Which forces a really practical question. Knowing how easily these hypersensitive tests can be confounded by an everyday heart med or kidney issue, would you want your primary care doctor interpreting these results, or should access be strictly gatekept by specialized memory neurologists?
This raised an intense debate regarding equitable access versus diagnostic accuracy, and it's one the medical community is actively wrestling with right now.
And they have to figure it out fast, because as we wrap up this deep dive into the source material, the undeniable takeaway is that the era of molecular phenotyping isn't science fiction anymore. It is here. We are no longer guessing at dementias through clock-drawing tests. We are tracing the exact molecular footprints of A-beta, tau, NFL, alpha-synuclein, and TDP-43 straight through our biofluids.
We are rapidly approaching a reality where a dementia panel won't be a dramatic one-time panning button when you're 75. It will become routine. Think of it like a PSA test for prostate health or a cholesterol panel for heart disease. It will be a sequential annual screening starting in middle age to monitor your brain's molecular trajectory over time, allowing us to guide targeted therapies before the physical scaffolding ever collapses.
But that leaves us with one incredibly heavy final thought to ponder. We are standing on the precipice of a world where a routine blood draw at age 45 could confidently tell you your exact molecular risk of developing a specific, devastating dementia at age 75. But until the medical field guarantees definitive, accessible cures for all of these misfolding proteins, do you actually want to know your brain's molecular fate? Or, when faced with an incurable timeline, is some ignorance truly bliss? Thank you for joining us on this journey into the mind. See you on the next deep dive.